Okay, if everyone could get seated, we're going to start now. Okay, all right, got to go ahead. I'm Mark Stein, and I just wanted to, thank you. I just wanted to say how thrilled I am to see everybody here, and we're opening up the APSARD meeting, which is just jam-packed with great things. Just a few housekeeping announcements. The program, the materials, we don't want people to be taking photos or tapes of them. Some of them will be available later. I guess the other thing I wanted to mention, if you didn't get the note, there's an organization called Citizens Commission on Human Rights, which might not be exactly that. Because of that, everyone should be kind of alert. You need to wear your name tags to get into the sessions. And if anyone approaches you, feel free to contact anybody from Parthenon in a green shirt. Also, from any house phone, you can dial 11. And if you're approached by anyone from the media, please also direct that to Parthenon. And Dr. Adler and I will be available to meet with them. We hope that's not a distraction. It doesn't take away from what we think is going to be a fantastic meeting. So anyway, welcome. I'm going to introduce Dr. Jeff Newcorn, the head of the program committee, who's going to get our program started. Okay, Jeff?
All right, thanks. Hopefully that's the last you'll hear about any of this stuff, and we'll just go on and have our usual excellent meeting. Actually, it won't be our usual excellent meeting. It will be an even better than usual excellent meeting, and we're really excited about that. I'll just tell you a couple of things about it. It's mainly going to follow the same format that it has before: plenary, symposia, parallel symposia in the afternoon, posters. There's just a couple of new things this year. First of all, the posters are mainly up now. We'll have the reception after this plenary in the poster room, but we are not asking the poster presenters to stand by their posters because we'd like them to have a chance to mingle with everybody as well, and we're asking them to do that tomorrow. So the posters will be there for viewing tonight if you want to do that, but you won't necessarily have the presenters there. Now, tomorrow during the poster session, we're also going to have a data blitz where 10 of the posters were selected for a brief capsule presentation orally. And the hotel layout is such that there's a room adjoining the poster room. And so we'll be able to have access going back and forth between it. It'll be the first hour of the poster session. So if you see something that catches your eye and you want to learn more about it, you'll be able to go to the poster, and then the presenter will be there, and you'll be able to have some dialogue and questions. Everything else is pretty much the same. We'll be doing a Lifetime Achievement Award this year in the morning tomorrow. We've cleared out time, legitimate time, for the Ask the Expert sessions that have been popular in the past, but were not well scheduled last year. We think it'll be a great meeting, and I welcome all of you. And I want to say how excited we are to have this plenary for our open plenary, and 3 outstanding presenters. And I'm going to let Phil Asherson introduce the session. Phil is a friend, of course, of APSARD. He's been here several times, and professor of psychiatry at the Institute of Psychiatry, King's College in London. And Phil, the podium is yours.
Okay, I'm going to go straight into the slides and introduce the whole symposia at the same time. Here are my disclosures. All payments I get go to King's College London. So I wanted to frame this session around the end of phenotype concept. And this is a diagram that you often see. It's often presented in grant applications. And you can see you begin with various different levels, thinking about the basic etiological factors, genes and environment, and then a range of different levels. How do those gene effects turn into cellular function effects? And how do those cellular function effects affect the structure of the brain, the function of the brain? How does structure and function turn into function, cognitive constructs, and then going on into behavioral impairments and clinical disorders like ADHD? And I think our talks probably begin at the bottom up. My own talk is going to focus predominantly on the area around what I call level 1, somewhere between behavior and a kind of cognitive construct and the link between that and neural function. Trevor Robbins, well, we'll see what he talks about, but I think it will be around psychopharmacology and brain function and how that impacts on cognitive constructs that could underpin conditions like ADHD. And Tony will focus mainly on neurotransmission, and particularly around the dopamine activity in the synapse around the dopamine neurotransmission. And I suppose all of these have in common trying to pick out mechanisms that can kind of track treatment effects, not only the clinical disorder, and make those links between treatment effects and clinical outcomes. And so they're very translational in the sort of underpinnings of these ideas. I've never been entirely happy with this diagram because what we actually know is that genes have many, many different effects. And so a lot of the things we measure in the brain are actually, in terms of causal, some sort of causal relationship to each other, they're often unrelated. They're kind of what we think of as pleiotropic effects. You know, genes have multiple different outcomes. And it's very hard for us to tell which of those outcomes links one thing to another. So I think focusing on treatment effects is one way that you can link things together. You can say, if I bring about a change in dopamine transmission, what impact does that have on neural function? And what impact does that change in neural function then have on cognition and behavior? And so it's one way of trying to link these things together. So I think in some way all of our talks will focus on these themes. So in terms of ADHD, I'm going to give a slightly new perspective where we think of neural dysfunctions as leading directly to what you could think of as a symptom, mind wandering, and that mind wandering in itself is like an endophenotype that leads on to the behavioral symptoms of ADHD. So ADHD and the wandering mind. So we'll just go one step back and begin by thinking, what is ADHD? Well, the DSM define it in quite a useful way. They say it's a persistent pattern of inattention and/or hyperactivity and impulsivity that interferes with or reduces the quality of functioning in daily life. And when you look at the symptoms in the DSM, what's always struck me about them is they're quite behavioral. They're more the kind of consequence and the outcome of some underlying problem that's leading to behavioral problems like difficulty keeping your focus or putting things down and losing them, getting easily distracted. But the question is, what is underlying those problems? The disorder is also defined by impairment. So we know that those, when you have high levels of ADHD symptoms, you're more likely to have problems in work, education, social life, coping, accidents, and so on. And those I think of as psychosocial functions. But you also get a lot of symptoms which overlap with mental health conditions, feeling distressed, low self-esteem, irritability, sleep problems. And so you get these different aspects to the condition. But some people like to kind of focus back on the idea that ADHD sometimes, some people have the ability to sort of take advantage of the fact that they have a wandering mind. Maybe that generates creativity. Perhaps the hyperactivity can be turned into a sort of useful, energetic outcome. And one question that comes up repeatedly from people working with adults' ADHD and adult mental health in particular is, how do we best discriminate the inattention of ADHD from the distractibility, the inattention of many other disorders? And I think I learnt something from Tom Brown here, which is that everyone with ADHD can actually do some things well. And so it's not really an attention deficit in the sense of a fixed deficit, like having low IQ, for example, but rather something which is rather malleable. And if you're doing something salient enough, you actually can focus. And so you get other characteristics in ADHD which may be more specific to the condition, like hyperfocus, ability to focus when when what you're doing is very salient and rewarding. People with ADHD get bored quickly and then lose their focus, but they focus, then get bored, and then lose their focus. And I rather like impatience when people are waiting. It's closely aligned to Edmund's idea of delay aversion. When people are waiting, they get very irritable, and you get this kind of what I call the ADHD shudder. Just thinking about waiting is enough to make somebody with ADHD feel irritable. But another aspect of the mental state is asking people about why are they inattentive, and what they often describe is excessive, uncontrolled mind wandering. So what is mind wandering? It's something that we all do at least half of the time. Mind wandering occurs when one mind drifts away from a task and focuses on internal thoughts that are unrelated to the task or situation at hand. That's one definition of it. It's universal. As I said, it occurs at least half of the time. But in ADHD, people describe their experience of their mental state in very characteristic ways. They use terms like, I'm constantly in a daydream, I'm in a fog, a whirlwind of thoughts, hamster on a wheel. My thoughts are like a jack-in-the-box, waves in a storm. It's like popping corn. And I've been collecting some pictures of this. This was a very early one that we had of a spaghetti head, reflecting this kind of distractible mental state. And one that I've always liked, where you've got 9 sort of volumes or channels, and they're all on full volume at the same time. You can imagine if you've got multiple different processes or lines of thought all together at the same time, it's going to be very internally distracting. I've been working with an artist who's been asking people to draw their mind wandering. And what the hell is that? Oh, that's just my mind. It's sort of rapid thought flow. My mind is a lot more interesting than real life. Well, could be. And some people really have a chaotic mental state. And this person said, the Mad Hatter, have I gone mad? I'm afraid so. You're entirely bonkers. So it can really make people feel that there's something wrong with them. So when we spent a lot of time talking to patients about this phenomenology, and we ended up thinking there were 3 main components to it. Thoughts on the go all the time, like a ceaseless sort of mental state. Thoughts jumping or flitting from one thing to another, and multiple lines of thought all together at the same time. So we made a rating scale, and I suppose the main thing to say about this scale is it's only asking about the mental phenomena. It doesn't ask about your behavior or the consequences, but just simply a description of this kind of mental state. And certainly in terms of discriminating ADHD cases from controls, it's really good at that. And what this is showing is that the Muse scale on the far left is about as good at discriminating ADHD from controls as the DSM inattentive symptoms, DSM hyperactivity and impulsivity symptoms. And you'll see on the right that actually emotion lability is also quite good at discriminating ADHD cases from controls. In terms of this mind-wandering measure that we developed, in light blue, you've got the correlations, cross-sectional correlations. And so you can see it's highly correlated to people's reports of inattention, hyperactivity, emotion dysregulation. And on the far right, functional impairment, using Margaret Weiss, who's in the audience here, but using her scale of impairment, it correlates very highly, 0.81 with functional impairment. In orange, these are actually correlations in the change in the symptom domains over a 6-month period. So what this is showing is that as your mind wandering goes up and down over a 6-month period, this correlates highly with changes in inattention within all the ADHD symptoms and impairments. And in particular, it seems to co-vary over time, mostly with inattention and also functional impairment. So when I first presented this, I remember Robert Plomin saying, well, what's the point? Because all you're measuring is it's just another measure of inattention. So why not just stick with the measures you've already got? And so we thought a bit about that, and at least we found one advantage in our analysis, which was its relationship to the Margaret Weiss's impairment scale. And what this was showing is that, first of all, we looked at the prediction of inattention and hyperactivity and impulsivity on impairment, which was very strong. But then when we added in mind wandering as an additional measure, it had more predictive value. It had an independent predictive value over and above inattention and hyperactivity. And if you put all three into the analysis together, you can see that the mind-wandering measure was the best predictor of impairment, followed by inattention and then by hyperactivity and impulsivity in this sample. So how can this distracting mental state to bring about behavioral and functional problems in ADHD? And I think you kind of think through a rather common sense set of arguments as to how that could occur. I mean, if your mind is constantly wandering off, it's going to be hard to follow conversations. If you're reading a page on the book, you're going to lose track of things. And it's very hard to sustain your focus when your mind's wandering off. It also is hard to hold thoughts in mind, which is a kind of working memory function, and then it becomes very hard to process things, think through things, and have strategic thinking and planning. And at a symptomatic level, people often describe feeling rather exhausted by the constant effort of having to work really hard to reduce mind wandering so they can focus, to keep focused, and to stop their mind constantly wandering off. People also talk about these symptoms keeping them awake at night. You know, trying to sleep, your mind is completely overactive. So at least it's sort of a sensible idea at that level. So the other problem, one of the problems with with mind wandering, of course, is it in itself is highly heterogeneous. As I said, it's something we all do all the time. And one of the key studies that started looking at this was Selly, who had rating scale measures of what he called spontaneous mind wandering and deliberate mind wandering. And what this study was showing is that in the relationship to ADHD, it was the spontaneous type rather than the deliberate type that predicted ADHD and that correlated with ADHD symptoms. So deliberate mind wandering, which is unrelated to ADHD, is much more to do with strategic thinking. You're driving your car and you're thinking about the grant, in my case, the grant you're going to be writing tomorrow. And that's not a problem. You can drive the car, think about the grant, and it's quite productive and strategic. And then if you need to focus back because somebody walks out in front of you, you're actually able to do that. You can switch your focus back quickly to the task in hand. That's very deliberate. It's a very controlled process. And it's very different from spontaneous mind wandering, which is really this kind of more like a random noise in your head that interferes with and stops you focusing. So maybe you're driving the car, somebody walks out, and you just don't notice them because your mind is elsewhere and you're not really sort of paying attention in that sense. So these models have become much more sophisticated, and one of the key papers in this area was by Christoph in her Nature Review article. And she essentially had two dimensions. One is along the horizontal axis, which goes from this being focused at one end to being unfocused at the other. And so this you can think of as a dimension of deliberate to spontaneous mind wandering, if you like. And the other dimension is much more to do with the content, actually, whether it's constrained, as in depressive rumination, where you have a repeated theme, and then at the other end, rather unconstrained, which is really more like ADHD. The thought content is on any old thing. And so ADHD really, we think, sits down here. And so there are few constraints in both of these two dimensions. And so what's interesting about these models is it's quite a sophisticated set of analyses that link different balances of neural regulation that sort of underpin where you are on these different dimensions. And they all seem to focus largely around these. I'm just trying to sort of really oversimplify things to try and get some coarse concepts here. But this came from a review on the salience network, and it rather nicely illustrates these different functions. The default mode, which has been linked very strongly to mind wandering, and then the central executive, and when you're focused, this is very active when you're focused. And the salience network sort of acts as a dynamic switch that pays attention to the environment, and depending on the salience of stimuli, will then sort of engage these processes, upregulating your executive to focus and downregulating your default mode, so that you stop mind wandering and can focus. And in fact, you can actually measure these things quite well, and even fairly simply, as in this video. And this is really, I think, measuring cortical activation. So as this child tries to focus, this monitor is picking up the EEG signal of cortical activation and is able to make this ball rise up in the air. It's quite interesting, the amount of variability in the ability to do this task among different people. So, what we know about mind wandering in general in controlled populations is it's very strongly linked to default mode network activity. And one of the very first studies that looked at this was from McKiernan in 2006. And what they did is they took a single task, an auditory detection task, but they manipulated the task with a range of different ways of making tasks more or less difficult to perform and requiring people to focus to varying degrees. And so you could manipulate the frequency of mind wandering under these different conditions. And then they looked at the relationship of the frequency of mind wandering to your ability to downregulate your default mode activity, what they call task-induced deactivation. And then you can see that across these 9 conditions, there's a very strong relationship between the frequency of mind wandering and task-induced deactivation, and the variance explained from this data set was about 0.81. So it's an incredibly strong relationship between these things. And then you can actually sort of ask in a somewhat more detailed way about the brain regions involved. And if you look at the frequency of task-unrelated thoughts and the regions where they correlate to task-induced deactivations, you end up picking out these regions which essentially are around the default mode network. So in these studies, they actually measured mind wandering outside of the scanner, and then they in a way repeated the studies on 2 occasions, measuring mind wandering on one occasion and then inside the scanner looking at brain function on another. So other research said, okay, but we really need to measure things at the same time so we can see exactly what's going on in the brain when you're focused on a task or when your mind has wandered off. So what they started doing was taking a sustained attention task, and around every minute or so they would interrupt you and say, well, are you focused on the task, or has your mind wandered off and are you thinking about something else? They also ask a secondary question, which is if your mind has wandered off, how aware are you? Are you aware of what you're thinking about? Or have you just completely lost your mind and you're not even aware of what you're thinking about? And doing a simple contrast between the periods just before somebody said they were focused or just where they said they were not focused, they were able to identify that there was activation of the default mode network in relation to this task-unrelated thoughts. But actually, interestingly, for the first time, they showed that there was executive control activation at the same time, and they were actually co-activating together during periods of mind wandering. The other thing they found, which was very interesting, is that if you then compared your mind has wandered off, but you know what you're thinking about, or your mind's wandered off, but you're not even aware of what you're thinking about, they're almost on a kind of severity scale. So your default mode is most active when you're completely unaware and not even aware of what you're thinking about. When you're aware of what you're thinking about, it's somewhat reduced. And when you're focused on a task, it's at its lowest sort of activity level. So there's a kind of severity scale, if you like. So these basic findings were looked at and then reproduced in a meta-analysis that actually had a lot of data that went into this study. essentially showing that both cortical control and default mode activity were both involved in mind-wandering processes. So it did start a debate about what's going on here. And I think it could be explained by these two different types of mind-wandering we already talked about, deliberate mind-wandering and spontaneous mind-wandering. So deliberate is where there's more executive control. And this would be reflected in increased strategic thinking, particularly under low cognitive demand situations like driving a car. And also mind-wandering increases in these low cognitive demand situations in people who have more executive resources, better working memory function. On the other hand, executive failure is really another situation where the executive control can reduce and suppress unregulated and uncontrolled mind-wandering. And so it's kind of a suppression of unwanted internally generated thoughts. And there you get higher mind-wandering in high cognitive demand situations in people with greater executive control functions. So I didn't want to go over time, because I'm the chair, I've left myself in a slightly awkward position. Could Jeff or somebody just remind me? Yeah, thanks, okay. The other thing that we thought was very interesting is when you start to look at different aspects of the control and the regulation of mind-wandering in healthy people and how a dysregulation of those functions could be reflected in ADHD, there seem to be a lot of parallels to draw between the two. And there are three main ones I'd like to just briefly focus on. One is something I've already touched on, which is context regulation. When a task is more difficult, you need to focus harder and your mind-wandering decreases. Salience is really the impact of reward. When something is more interesting, you focus more and your mind-wandering decreases. And the other one is sensory decoupling, which was a finding that when your mind has wandered, you actually often miss things at a very early sensory level. So it's not just about cortical control. There seems to be a sensory deficit when your mind has wandered. So beginning with context regulation, in this study we had a very simple measure of cortical activity, which is a midline frontal theta. And this diagonal black line is showing the effects in healthy controls. First of all, in a resting state condition, and in the middle, doing the CPTOX, which is an easy task, and then finally the SART, which is a much more difficult task. So as you go from rest to an easy task to a difficult task, you get increase in cortical activation. And this is the ADHD group, this horizontal line. So you can see they're really failing this appropriate context regulation. And so here you can look at this before treatment, the control group in blue, and then the ADHD group in yellow. And then when the ADHD group are on methylphenidate, it actually normalizes this context regulation. Another study that showed this in a different way was from Katia Rubia, where she manipulated the presentation rate. And I call this the slow and boring task, because as you increase the time gap between stimuli from 2 seconds to 5 seconds to 8 seconds, some people find it increasingly hard to keep focused when you have to wait 8 seconds. And of course, there's a lot more time for your mind to wander off. And so in healthy controls, looking at function within the central executive, you see that you get an incremental increase in activation as the task gets more difficult. But here in the ADHD group, you can see that's completely lacking. And on the other hand, if you look within the default mode, in the healthy group, you can see the deactivation of the default mode. As you need to focus, you have to deactivate your default mode. We assume, although it wasn't measured, that your mind wandering would go down. And here, on the other hand, in the ADHD group, you get a completely different effect. And if anything, your default mode becomes more active, at least in this particular data set. So ADHD and controls are very different in relation to context regulation at the neural level. So the other thing I talked about was sensory decoupling. And this really was shown very nicely by Baird et al. In Healthy Controls, doing a contrast between people when they were focused on a task and between people when their periods when their mind had wandered off. And so the top image here is looking at somebody who's focused on the task, and the bottom image is showing what's happening when their mind has wandered off. And this yellow flare is really showing neuronal activity that's occurring it's really the timing that I wanted to emphasize, that this flare is occurring at around 100 milliseconds after the stimulus. So this is a very early sensory response, somatosensory response. And you can see the difference here in the ERP, and this is actually sitting over the occipital cortex. So it's a kind of early sensory deficit. When your mind's wandered, your brain isn't responding to sensory stimuli in the same way. There have been less studies of this sort in ADHD, but there was a very nice study from Rosemary Tannock actually looking at sensory stimuli. And if you stimulate the peripheral nerve, she found a reduced early sensory response to the stimulation of a peripheral nerve in ADHD compared to controls. In our own work, we actually looked at what was going on before and after errors on the assumption that when you made a correct response, you were focused, and when you made an error, it may well be that your mind had wandered and you were unfocused. And again, doing those comparisons within ADHD, we found that there was a greater sensory signal prior to a correct response and a reduced sensory signal prior to an error. So I think we need to do more work on it, but it does seem from this and other studies that there are sensory deficits in ADHD, and they match those seen during periods of mind wandering in healthy controls. So finally, I wanted to talk about the effect of reward. And we've known for a long time that both in terms of cognitive performance and also your ability to focus in daily life, that when you're doing things, when things are under a more rewarded condition, there's this kind of catching up. And so in terms of reaction time variability, for example, we showed that in a slow unrewarded condition, there were bigger case control differences than when you were rewarded. And one of the key studies that has looked at this was from Elizabeth Liddell, looking at the activity within default mode, on and off methylphenidate, and under rewarded conditions, and rewarded and unrewarded conditions. So what this is showing here is that these children are doing a go-no-go task. It's a very boring task, and under a low incentive condition, off medication, what this is showing is their default mode is relatively overactive compared to the controls. When they go on medication, it actually normalizes that, and they're now able to deactivate their default mode. What was interesting is when they repeated the study but rewarded people while they were doing the task, they made the same task, but they made it rewarding and more interesting. This actually was able to, they were now able to deactivate the default mode even when they were not on medication. And they made this general conclusion that in this study, methylphenidate and reward had the same effect on normalizing neural function in ADHD. So it comes back to really thinking about these different processes. And if we're trying to understand what might be going wrong in ADHD, of course, these types of deficits could be due to abnormalities sort of really anywhere within this circuitry, I think. And so one would have to do a lot more work to try and sort of pick out whether it's really a problem with executive control. Is it a problem in the salience network? Is it, what else could be affecting default mode regulation and the interaction between all these things? And we certainly know that in ADHD there's reductions in connectivity across these various networks compared to healthy controls. So just a few more words and then I'll stop. But the research seems to be leading away from what was a very strong hypothesis, where the primary deficit was thought to be top-down executive control dysfunctions leading to ADHD, and maybe perhaps moving more towards saying there's some aberration in the interconnections between the salience network and the default mode network. And this was one of the findings from Castellanos, with Sanugabat was also, and Len Adler and others were on this paper, showing that there was perhaps a new locus of dysfunction, linking the cingulate and the precuneus interactions between the salience and the default mode network. In our own work, we took another approach, which was focusing on cognitive functions, but looking at people, following up children with ADHD into adulthood and looking at those that persisted and those that remitted from ADHD over time. And we'd identified that there were these separate functions, reaction time variability as a cognitive measure, which was very malleable and very sensitive to reward, and executive control functions like inhibitory control and working memory, which were much more stable and fixed and insensitive to salience and reward. And essentially we found that it was the measures linked to reaction time variability, what we called preparation and vigilance measures, that tracked the clinical disorder over time and were seen in persistent cases of ADHD but not in remitted cases of ADHD. Whereas the executive control measures, like working memory and inhibition, they were found in cases of ADHD at outcome, whether their ADHD had got better or not. So there was a kind of dissociation of ADHD from these executive control measures through development, and one no longer predicted the other at outcome. Similar findings were actually found from a group here, from Biederman and colleagues' group, using resting state data, and again looking at persistence and remission of ADHD, and showing that it was really measures linked to default mode network activity that tracked the clinical disorder over time, whereas measures linked to executive control functions were persistent, but they were found in both persisters and remitters. And so they, again, dissociated from ADHD through development and no longer predicted the clinical disorder at outcome. So overall, we think that these perhaps refocus where we think these dysfunctions might lie. But in particular, if you can target the regulation of the default mode or the interactions between the salience network and the default mode, they may be the best targets for treatments. So just to wind up, we've slightly reframed our model of ADHD. We think, of course, there are brain dysfunctions. We think that they may primarily lead to inability to keep focused on a task and generate this rather uncontrolled, spontaneous mind-wandering state. And that in itself is enough to generate many of the behavioral symptoms of ADHD. So I'll end on that slide. We're very interested in the effects of medication on these systems. We're wondering about things like mindfulness. You know, meditation does seem to target these same processes. And although I was rather skeptical about mindfulness, I was quite impressed by this publication from a Dutch group showing in a randomized control trial effects as good as stimulants from mindfulness. So I remain skeptical, but I think it's very interesting. So I'll end up with that. Some people say mind wandering for people with ADHD is both their best friend and their worst enemy. Thank you. We probably don't have much time for questions. Shall we go straight on to you, Trevor?
Well, thank you for this great honour of being invited to this exciting plenary symposium. My disclosures are listed there. And I want straight away to say that a lot of our research has been informed by this brilliant conceptual framework, which I think is still highly relevant, which links genetic changes in terms of etiology and epigenetics all the way through to the diagnostic heterogeneity of ADHD, including these key symptoms, of course, of hyperactivity, inattention, and impulsivity. And I'm going to focus on the, if you like, the mediating neuropsychological constructs associated with ADHD, which include various aspects of executive function, inhibitory response control, the monitoring of attention, and the updating of working memory. And possibly underlying substrates, which include, as you see here, very presciently in this review, the right frontostriatal hyperplasia and hyperfunction hypothesis, about which I'm going to say quite a good deal. Also catecholamine dysregulation, by which I mean dopamine and noradrenaline, and for which much of the evidence derives from the therapeutic effects of stimulant drugs, Ritalin and amphetamine. Now, of course, these drugs are among perhaps the most successful, if one can say that, of all drugs used in psychiatry for treatment. And a variety of trials has attested to the efficacy, certainly in severe cases of ADHD, of conventional stimulant drugs, amphetamine and Ritalin, and also non-stimulant drugs such as atomoxetine or Strattera. And atomoxetine has a more selective action than the methylphenidate and amphetamine. So as you know, both methylphenidate and amphetamine block the dopamine and noradrenaline transporter molecules, thereby increasing the amount of catecholamine in the synaptic cleft that's available for stimulating postsynaptic receptors. But of course, I should also remind you that there are inhibitory autoreceptors on the cell bodies of these neurons, and oral or injected stimulant will also affect those receptors as well. So the net action of these drugs depends on a balance between the inhibitory autoreceptor action and ultimately the postsynaptic action. And this is something that Tony Grace, I'm sure, will discuss in more detail. But the big question I have for you, and for everyone really, is, okay, these drugs affect both dopamine and noradrenaline. So which one of these neurotransmitters is most responsible for the therapeutic effects? Not many people think about this, it seems. And where in the brain are these effects located? Can that give us some idea about how one dissects the cognitive deficits in this disorder? And I'm going to try and attempt to answer this question, but I'll tell you what the provisional answer is. I think the drugs affect both neurotransmitters in several different parts of the brain to produce their optimal effects. Now, atomoxetine does not block the dopamine transporter, and so it has a much more selective action on the noradrenaline system. So one can gain insight into this issue by comparing the effects of stimulant drugs and non-stimulant drugs such as atomoxetine. Now, I should also remind you that these drugs are working within the context of what we call the frontostriatal networks in the brain. Here is the human reward system, the head ganglion in the frontal cortex, and the nucleus accumbens or ventral stratum, an important way station. And it's shown here by the hot loop, modulated by dopamine in particular. And these loops, these functional loops, are reproduced in different sectors of the frontal lobe and are undoubtedly relevant for the control of executive function. Now, most of our work is on adult ADHD, especially because we have studies using positron emission tomography. And I just wanted to make the point that these patients, these adults, of course, had a diagnosis originally of juvenile ADHD, and they also have severe executive problems, and added to that, they have structural loss of grey matter in relevant brain regions. So, for example, in this study, we showed particular reductions in the right inferior frontal gyrus, also the left middle frontal gyrus, as well as in the hippocampus, amygdala, limbic structures, and the cerebellum, and also changes posteriorly in the putamen. So this is a classic frontostriatal disorder, but there are changes in other regions. Now, following up those data, I just wanted to inform you about a study looking at the endophenotype, if you will, for adult ADHD. In this study by Pronti and others, which is an fMRI study, and also looking at structural changes as well, using a task which probably severely stresses mind wandering. This is a sustained attention task where digits are presented at the rate of 100 per minute in the central location, and you have to detect sequences which are defined here. So the task really severely stresses sustained attention, and it has a working memory component. Now, both adult ADHD individuals and indeed their non-affected siblings have severe deficits in this task as measured by the signal detection parameter A prime. Intriguingly, this is somewhat related to a significant cluster of decreased grey matter volume in the right inferior frontal gyrus in both of these groups. And so this was the major test in our extensive battery, which looked at many other functions, looking at these individuals. And this is the main deficit we saw in the relatives, this impairment in sustained attention, also paralleled by the change in structure in the inferior frontal cortex. Now, in our PET study of these patients, we used D2 binding as a measure with falapride as the ligand. And of course, one can beautifully identify D2 receptor binding in the stratum using this method. But we also, of course, challenged these subjects with methylphenidate in order to show the reduction in binding which occurs following this drug. The reduction in binding is because of the increase of dopamine release and the presence of dopamine in the synaptic cleft, which displaces the ligand from the receptor. And so one can use this to measure basal D2 receptors, but also, to some extent, dopamine release or dopamine regulation following the stimulant drug. Now, there have been a number of other studies looking at this, and it's a somewhat tricky literature. Probably the biggest finding in the largest population that's been reported so far is a reduction of D2 receptors in the left caudate nucleus in an important study by Volkow, which used a very large number of ADHD individuals. We had a smaller number, and I have to report to you no changes in D2 receptor binding throughout different sectors of the stratum and also in the midbrain. You can see here no really significant changes at all. As I said before, Volkow saw a small change, rather a low level of significance, in the precommisural caudate nucleus. Okay, but it's interesting to note that nevertheless, if one looks at correlations with the ADHD self-report scale, both in the normal controls and in the ADHD individuals, you do nevertheless see significant relationships with D2 receptor binding. It's just that the ADHD individuals have much higher levels of symptoms, of course. So it's interesting that this is related in some sense to dopamine changes, but it also perhaps highlights the fact that maybe adult ADHD is in some sense on a continuum of deficit, which you may even see to some extent in the normal healthy population. This is a hypothesis we've entertained before. Now, when one looks at the effect of Ritalin on sustained attention in this task, we didn't see significant effects, although there were trends. However, we did see improvements if one segregated the sample into high performers and low performers. Now, the low performers contained a very large number of the adult ADHD individuals, but also some poorly performing control subjects. So again, that stresses sometimes the spectrum nature of this deficit. And you can see Ritalin improving performance here. When one looked at changes in D2 receptor binding as a function of this, you can see under placebo, indeed, the low performers have lower D2 receptors in precisely the same region of the stratum that Volkow saw her effects. A significant reduction in D2 receptors there. And after Ritalin, the receptor binding is reduced in both cases, although it is higher reduction in the high performers. And intriguingly, we also, of course, measured D2 receptors in the midbrain, where the receptors are auto-inhibitory receptors on dopamine cell bodies, regulating release. And again, we found a much bigger change in the high performers compared to the low performers after Ritalin. So this may suggest some difference in the way dopamine is regulated in these nigrostriatal and ventral tegmental to nucleus accumbens projections. Well, if we try and delve further into this, we probably need animal models, and there are a number of animal models etiologically of ADHD, including genetic mutants, mainly in mice, rodent strains, lesions, early stress. But I'm simply going to focus on what we see in the normal population of healthy Lister-hooded rats, looking at poor performers and high impulsive rats. And for this purpose, we use a task which measures both sustained attention and impulsivity in the same paradigm. It's derived from a human test of continuous performance called the serial choice reaction time task. And in this test, the rat scans an array of apertures to detect briefly presented visual stimuli associated with food reward, which are presented in random at one of these locations. If the rat correctly nose pokes in that location, it receives a food reward and then goes to the next trial. There will be 100 trials in any session. If he responds in the wrong hole, he's plunged into darkness as a punishment and has to go to an empty food magazine to continue the task. Additionally, if the rat responds prematurely, in other words, tries to outguess where the computer places the next visual stimulus and responds in advance of that stimulus prematurely, it's also punished. And we classify this operationally as a premature response, an impulsive response. Now, the rat uses virtually the whole of its frontal lobe and striatum to perform optimally on this task. And we've shown this in a number of ways, using lesions, for example. And we know that the accuracy of the rat is controlled by the anterior cingulate cortex and its frontal cortex. But intriguingly, a different area in the ventral medial prefrontal cortex, in the infolimbic region, controls impulsivity. So rats with damage there show impulsive responding, whereas rats with damage here show impaired accuracy. And these effects are clean. In other words, you don't get the other symptom associated with each lesion. And as I said before, these regions project into the stratum. This is the rat stratum, the dorsal stratum, the ventral stratum, the nucleus accumbens. And you can see that damage in the nucleus accumbens also induces impulsivity. Now, intriguingly, if one modulates dopamine function in this region, in the rat, in the medial prefrontal cortex, in low-performing rats, that's to say rats who are inefficient at detecting the light and show lots of errors, a D1 agonist, a D1 dopamine receptor agonist, will improve performance up to the level of high-performing rats. So here's some grounds for believing that Ritalin might well affect this type of mechanism in the normal animal and the low-performing animals. Now, focusing on impulsivity, we find that about 10% of our rats show a consistent trait of responding prematurely, as shown here in this bimodal distribution. These are high impulsives. Here's an example of a high impulsive rat. His name is Zippy, and we've used Zippy in extensive breeding programs to try and look at the genes associated with impulsivity in the rat. And in our GWAS study, we find genes on chromosome 1 associated with GABA and glutamate function, as well as some synaptic proteins. But also intriguingly, in our high impulsive rats, we see a reduction of D2 receptor binding, measured also with falipride, in the nucleus accumbens, in the ventral stratum, compared to the low impulsive rats. No changes in the dorsal stratum, but significant reductions here which correlate with the degree of impulsivity. So we do see changes in D2 receptors in these high impulsive rats. And also intriguingly, if we allow these rats to self-administer drugs of abuse, such as cocaine, through an intravenous catheter, they will self-administer more. So these high-impulsive rats will self-administer more cocaine. Now, it's intriguing that this act actually treats their impulsivity, because if it's tested at the same time, these rats become less impulsive. So that's encouraged us to do some studies. This is with Jeff Daly's team, looking at the effects not only of self-administered cocaine, but also of oral subchronic Ritalin, or methylphenidate, on impulsive responding. And this is what we find in the pre and post stage of Ritalin with high impulsive and low impulsive rats. So high impulsive rats show reductions in impulsivity with Ritalin, just as you'd expect. Low impulsive rats show increases in impulsivity. And this is entirely consistent with what we see with other dopamine stimulating drugs. So there's a baseline dependency here, which explains in a sense the paradoxical effect of Ritalin and amphetamine in these animals. Now, associated with this are changes in the D2 receptor, and it works in the same way. The high impulsive rat with low D2 receptors, and I should say this may also be D3 receptors, it's not quite clear, these animals have elevations of receptors after a regimen of subchronic Ritalin, so it upregulates the D2 receptor, whereas the low impulsive rats who have higher D2, D3 receptors have reductions, which makes them more impulsive. So there's an intriguing baseline dependency neurochemically as well as behaviorally. But when you do the appropriate statistical analysis to see how much of the variance this effect is responsible for, you find in fact that it's only responsible for a fraction really of the total variance. And that leads us to believe that noradrenaline may also have a role in regulating impulsivity in these animals following Ritalin, because Ritalin, remember, will increase noradrenaline levels as well. And we've got some evidence for this on the basis of the effects of atomoxetine. So atomoxetine, as you see, dramatically remediates impulsivity in these rats. It doesn't affect attention very much. It's not a sedative drug. And when the drug is actually placed into the nucleus accumbens itself by intracerebral infusion, it produces the same effect. Whereas if one blocks the reuptake of dopamine in the structure, you get increases in impulsivity. So increasing noradrenaline in the shell improves impulsivity. And this is probably due to subcerebral and locus coeruleus projections to this sector of the basal ganglia, which is the only sector which receives noradrenergic innervation. This defines some of the brain networks associated with this premature responding or waiting impulsivity, including the top-down control mechanisms that I mentioned earlier, including the infolimbic cortex. But intriguingly, this region we've also shown is implicated in another form of impulsivity, namely temporal discounting, where one chooses a small or large reward as a function of delay. And here, as you'll remember, impulsivity is shown by taking the small immediate reward when given the choice, rather than being patient and waiting for the long reward. And we've known for many years that damage to the nucleus accumbens impairs this behavior, so they become highly impulsive. This is damage to the core region of the accumbens. And intriguingly, our high impulsive rats, higher premature rats, also discount reward more steeply. So they're impulsive on this count as well as being premature. Okay, and atomoxetine, intriguingly, in these rats also improves their performance, makes them more patient, and so they will tolerate larger delays for reward. Okay, so moving to humans now, looking at impulsivity, we've used a number of tests, but I want to introduce a test we've also used in rodents called the stop task. Now, in this test, it's a very easy task where your responding is governed by a computer presenting commands to respond on the right button or the left button, repeatedly, very quickly in a number of trials. But every 5 trials or so, every 20%, a beep may sound, which signals that you must cancel your response on that trial. You mustn't respond. By modelling the response time distributions, it's possible then to get a measure not only of go reaction time, but also of stop reaction time, the ability to inhibit your behaviour. And this task very successfully measures deficits in adult and juvenile ADHD, as you can see here. And this is the remediating effect of Ritalin in adult ADHD and also of atomoxetine. So both of these drugs improve performance in adult ADHD. In terms of the neural network, Adam Aaron, who did some of the original work with us, has mapped a neural network which includes not only the basal ganglia and the supplementary motor area, or the pre-supplementary motor area, but this very important inferior frontal cortex region, particularly on the right-hand side. And this network is associated with increased stopping. Now, our studies in rats, because we also have a rat version of this task, as well as humans, suggest that performance on this test is mediated not by the same network as the premature response or the discounting, but by a parallel network which devolves around the dorsal stratum, which includes the caudate and putamen, where it's modulated by dopamine there, but also by these top-down structures, including in humans, the right inferior frontal gyrus. In rat, this turns out to be the orbitofrontal cortex. Now, just to highlight the role of this right inferior frontal region, here's a functional MRI study where we compared stopping and shifting in a complex paradigm. And we found that, first of all, in general, much more activation is associated with stopping in the right inferior frontal cortex than shifting, which suggests that it's more involved in inhibition than in attention per se. But in ADHD, as you can see, this is adult ADHD subjects, they have less activation of the right inferior frontal cortex in this region associated with impaired stopping performance. So again, this highlights the special role of this structure in this network. Further evidence has been provided by a developmental study of Gunther Schumann and others in the so-called Imagen project, which was a collaboration coordinated by the Institute of Psychiatry over 10 centres in Europe, where we assessed 2,000 14-year-olds recruited from schools, healthy individuals, but then subjected to neuropsychological assessment, whole genome analysis, and brain imaging, including fMRI. Now, I'm just going to show you a very small snapshot of this study, but this is the stop signal performance in, at this stage, 1,250 of the 14-year-olds, using factor analysis to parcel out 7 orthogonal neural networks underlying impulse control. And in terms of ADHD symptoms, remember these are healthy individuals, but some of them did have ADHD-like behaviours associated with impaired go-no-go performance. You can see that those reporting ADHD symptoms had reduced activity in a bilateral right frontal error monitoring network. So this also occurred in the left frontal and in the basal ganglia on stop-fail trials only. So it's interesting that, again, this network is highlighted. It's also interesting that, genetically speaking, superior performance was associated with alleles of a polymorphism of the noradrenaline transporter, as you see here. Now, why this is relevant is because of studies we've undertaken subsequently, led by Sam Chamberlain, involving some collaboration with Katia Rubia, on the effects of atomoxetine in healthy individuals. First of all, intriguingly, you can see that atomoxetine improves stop signal reaction time even in healthy individuals. Again, underlying my point about the way these drugs seem to improve performance sometimes in normal subjects, whereas citalopram at doses which affect other functions has no effect on stop signal reaction time. And this improvement is associated with increased blood flow, again in the right inferior frontal gyrus. And moreover, this is associated with successful inhibition when you associate this with the plasma levels of atomoxetine in these individuals, whereas failed inhibition, there's no relationship. So in other words, using genetic, imaging, behavioural evidence, it does seem that noradrenaline is modulating this right inferior frontal network, including obviously the striatal outflow, in stop signal performance. Well, how about translating this to other animals to try and find out in more detail what the neurochemical basis of this? And we find in the rat, in our stop signal tarsal rat, that we get similarly, again remarkably, at exactly the same doses that we've used previously, significant improvements in stop signal reaction time and no effect on other aspects of performance. So this drug, therefore, improves premature responding, delayed discounting, and stop signal performance when given systemically. And you'll remember that the effect on premature responding depended on its effect in the stratum, in the ventral stratum, rather than the frontal cortex, for example. However, for stop signal reaction time, the effect is different. We find when infused into the cortex that this drug highly significantly improves performance in the orbitofrontal cortex, the suprarhinal region, which is probably homologous to the inferior frontal cortex in humans. There is also an effect in the cingulate cortex, which may be associated with the attentional component of that task. Okay. Additionally, Adrienne Barry, who did this experiment, showed with a number of pharmacological manipulations that this effect was noradrenergic. It wasn't caused by the fact that atomoxetine can increase dopamine in the cortex because dopamine is cleared there by the noradrenaline transporter. But it's not dopamine, it's noradrenaline. Okay, so here's my summary. My summary is that these performances are mediated by frontostriatal loops. Here are some of these loops reimagined from what I showed you earlier. I've shown you some evidence that sustained attention can be measured in this paradigm and depends on the anterior cingulate cortex in the rat and to some extent the dorsal stratum and the nucleus accumbens. Whereas impulsivity, we have three different forms: premature responding, delayed discounting, and stopping. And each of these seems to involve a subtly different striatal network, involving different sectors of the stratum, and differential modulation to some degree, although it's interesting that atomoxetine improved performance in each of these. Now, why I think this is theoretically interesting is that given the heterogeneity of ADHD, it seems to me from studies by, for example, Salantos and Ugo Bark, where they've looked at these different measures of impulsivity in ADHD children and shown not a very high degree of correlation. That might very well be because they're mediated by different networks, and so subtle differences in the spectrum of neural changes occurring in these disorders may relate to this heterogeneity. And of course, because these systems do project to all of these regions, there is the possibility that their functions can be titrated optimally by appropriate dosage. But this will be not only dopamine, but also, as you see, noradrenaline. I should also say that dopamine changes probably are much more important for the improvements in working memory we see in these procedures. Okay, it leaves for me to acknowledge my collaborators, in particular Jeff Daly and Adrienne Barry and Dawn Eagle for the rodent work, and a whole range of clinical collaborators led by Barbara Sahakian and including Natalia Del Campo and Visconti Pronti. Thank you very much. I think we better go straight on to Tony.
I think the way we've started, and we'll keep all the questions together at the end. I should have, I think after my talk, I did forget to introduce you, Trevor, and one thing I noticed was you've been in Cambridge since 1975, is that right?
I just want to tell you guys that, if we go over, you started late, we don't want to penalize this plenary. Certainly don't want to penalize Tony, and we want to have time for Q&A. If we go to the 7:45, it's okay. And if it's okay with you guys, okay. Those that are here want to hear this stuff, right? And I've been assured that it won't impinge on our ability to have the reception, that we can push that back too. So go forward.
Yeah. Yeah, I was noticing you got your PhD in 1975. I was just reflecting that I was 15 at the time. So I think it's absolutely tremendous, the work that you've done in Cambridge all these years and are so active in the field. So thank you, and the great experience in this panel. And so equally with Anthony Grace, who's Professor of Neuroscience and Psychiatry, a distinguished professor in Pittsburgh, and for many years has done translational research related to the dopamine system. So it's really great to hear you think about the more detailed dynamics and thinking about that in relation to ADHD. And, and we will have a discussion afterwards. Thank you.
If I had known that, I would have left those last 30 slides in. Anyway, what I'm going to be talking about is some of the work that we've been doing, looking at dopaminergic systems, how they interact with the circuits that Trevor has been talking about, and how we think it may be related both to the pathophysiology of ADHD as well as the treatment with stimulant drugs. Now, as Trevor had mentioned, the nucleus accumbens is really an area that we have a lot of interest in because it really is an area of integration, the ventral striatum, as far as its involvement in reward, its involvement in locomotion, and also its involvement in attention. And it's an area that integrates information from a number of cortical and limbic systems. The nucleus accumbens has a very powerful input from the prefrontal cortex, especially the medial prefrontal cortex, such as the prelimbic and infralimbic that Trevor had mentioned, that, among other things, is involved in behavioral flexibility. It also gets a very potent input from the more limbic aspects of the hippocampus, areas that are involved more in contextual focus and keeping you focused on task. And we think it's the interaction between the flexibility and the focus on task that's responsible for normal behavioral actions to be optimized, and also deficits that we think occur in ADHD. So in ADHD, patients exhibit disrupted and focused attention and instead tend to shift focus and interrupt their ability to work towards a goal. What we believe is that in this condition, the prefrontal cortex, which should only be shifting attention when a strategy is ineffective, is now inappropriately shifting strategies before a goal can be achieved. And this can happen by a disruption of hippocampus accumbens contextual focus. Now, how do the prefrontal cortex and hippocampus compete for control of goal-directed behavior? Well, in order to do this, what we wanted to do is to look at how the prefrontal cortex and the hippocampus interact in the nucleus accumbens. And to do this, we electrically stimulate these areas. And to look at the impact of the terminals in this area, we put a field recording electrode that allows us to record these field potentials that give us averaged input that's coming from each of these areas after stimulation. And from this, we can look at plasticity and also interaction between these systems. Now, what we find when we look at the hippocampal input is if we repetitively stimulate the hippocampal input, it potentiates the hippocampal input, a classic LTP, or long-term potentiation. But if we then follow it with a prefrontal cortex stimulus, it actually produces the opposite effect. When we look at the prefrontal cortex, hippocampal activation decreases prefrontal input, whereas prefrontal stimulation reverses that. So what we have are opposite actions, where whenever we activate the hippocampal input rapidly and repetitively, you get a strong potentiation of the hippocampal input and inhibition of the prefrontal input. On the other hand, if we do the same thing with the prefrontal input, it now inhibits hippocampal input and potentiates prefrontal input. So what we have is a competition that's occurring between these systems. When the hippocampus system is activated, it seizes control and maintains focus. But whenever the prefrontal cortical input is activated, it removes that hippocampal contextual focus and allows flexibility. So again, high-frequency activation of the hippocampal inputs potentiates the hippocampus and inhibits frontal cortex, resulting in increased focus on task, which is what we want. But on the other hand, high-frequency activation of the prefrontal inputs potentiates the hippocampal inputs and inhibits the hippocampal inputs, meaning that now you're going to start to get shifting from the tasks. So these, as I mentioned, these systems compete with hippocampus focusing on task, medial prefrontal cortex shifting focus. Now, the ventral hippocampus, in addition to allowing focus on task, drives context-dependent phenomenon. And as I mentioned, the prefrontal cortex allows the system to break out of the context. So if you're doing something, you're maintaining focus, but it's not achieving the type of goal that you want, the prefrontal cortex has the ability to come in, break that attentional focus, and allow you to shift to something that's going to be more behaviorally effective. But in ADHD, this system is disrupted. And instead, the ability to focus on a task to achieve a goal is disrupted. And one possibility is that the prefrontal cortex is inappropriately shifting behavioral strategies before it's appropriate to do so. The hypothesis is that psychostimulants enable the maintenance of the focus by shifting the balance by modulating the dopamine system, and particularly in what we call tonic and phasic dopamine systems, which is a dynamic regulation of the dopamine system that seem to achieve different behavioral outcomes by modulating these areas differently. Now, in order to do this, what we do is we record from dopaminergic neurons. Now, we know from a number of studies that just recording from a single dopamine neuron doesn't give you a good picture of what's going on, because we know that only about half the neurons are spontaneously active. The others are more in reserve and can be activated when needed. Furthermore, the ones that are firing at different firing rates and firing patterns. Now, in order to assess what's going on in the system, what we do is pass an electrode through this ventral tegmental area in a preset pattern. And as we're passing the electrode, we count the number of neurons that are active in each electrode track. And this is something we call population activity. We then look at the neuron to see how fast it's firing and also its firing pattern. Now, we look at these three parameters because they're all controlled by different circuits and have different functional impacts on behavioral outcomes. So what we find is that whenever we look at the number of neurons firing, this seems to be very potently regulated by the pathway starting in the hippocampus, the ventral hippocampal area called the subiculum, the area that's involved in contextual focus. If we activate this area by infusing NMDA, what we find is essentially a doubling in the number of neurons firing. So the population activity, the number of neurons firing increases without changing firing rate and firing pattern. So what we've done is to increase the tonic dopamine system without affecting the rapid phasic system. We know it's working through this pathway because we can actually interrupt this either by giving a glutamatergic antagonist into the accumbens, which completely blocks this, as well as by manipulating the ventral pallidum. So what we have is the ventral pallidum is a GABAergic area, fires in very high frequencies and very potently inhibits these dopaminergic neurons. When the hippocampus is activated, it activates the nucleus accumbens, which inhibits the pallidum and causes this whole population of neurons to start firing at their basal firing rate and pattern. This tonic dopamine release is actually a very low-level release. It occurs with slow, steady firing of the neurons. It depends on the number of neurons firing. It can be modulated by different kinds of inputs and contributes to the extracellular concentration of dopamine. Now, this is important because the extracellular concentration of dopamine, although it's a fraction of the concentration that you see inside the synapse, plays a very important role. It's on the order of about 10 to 50 nanomolar, depending on population activity. But it's potent enough to hit presynaptic receptors on dopamine terminals and presynaptic receptors on frontal cortical input. So it has a very potent ability to modulate inputs that are coming into this area. Now, in addition to this number of neurons firing, another important variable is firing pattern. Dopaminergic neurons at their baseline state fire in a slow, irregular type of firing pattern. But if an animal is exposed to a behaviorally salient stimulus, what you find is the neurons fire these bursts of action potentials. This is the phasic dopamine response, the rapid response that occurs whenever a behaviorally salient stimulus needs to be attended to. Burst firing basically alerts the brain that an important event has happened, something that you need to focus on and pay attention to. Now, this is controlled by a different input, a glutamatergic input coming from the pedunculopontine tegmentum, a brainstem nucleus. If we activate this pedunculopontine tegmentum, what we get is a very different response. Now we get an increase in burst firing, the phasic response, without any change in the tonic response. So the glutamatergic input, because of the way it works, is only going to cause neurons that are already firing to burst fire. This is because neurons that aren't firing, they're hyperpolarized, there's a magnesium block of the NMDA channel, and they're not affected. So only the firing neurons can show this phasic, rapid, behaviorally salient response. Again, the burst firing now leads to a massive dopamine release. As opposed to the 10 to 20 nanomolar concentration of dopamine extrasynaptically, within the synapse, it's hundreds of micromolar to millimolar concentrations. But it's also very rapid, so even though it's going to come out and completely saturate all of the receptors in this area, it's going to be very rapidly removed by reuptake before it can escape the synaptic cleft. So what we have is a compartmentalization of the dopaminergic system. The phasic synaptic release is a massive dopamine release that's caused by burst firing. It's fast acting because it's limited by the dopamine transporter, and you can't detect it with traditional neurochemical measures like microdialysis. But because it hits these D2 receptors inside the synapse, this is what you see with raclopride displacement. In contrast, the extrasynaptic or tonic levels are regulated by the number of dopaminergic neurons firing, activating these terminals. It would influence large ensembles of striatal neurons, and especially presynaptic terminals, by volume transmission. It's much longer lasting because it's now outside of the area where the dopamine transporter is going to have a big effect. And it's detectable by microdialysis, but not by something like raclopride displacement. And this is not heavily influenced by reuptake. And this is modulated by the pallidum. So the baseline dopaminergic neuron population activity determines the low-level tonic dopamine release. But when it's activated into burst firing, you get the behaviorally salient rapid phasic dopamine response that's usually associated with reward, but can also be associated with other types of salient stimuli. Now, what's the role of this tonic and phasic dopamine transmission when it comes to controlling the inputs coming into the accumbens? Well, what we do is differentially activate tonic and phasic dopamine by activating these two pathways that control these systems and see how it impacts afferent stimulation. So again, we stimulate prefrontal cortex or hippocampal inputs, we record local field potentials, and now we can micro-infuse drugs right next to the area where we're recording in order to see the dopamine contribution to the type of phenomena that we see. Now, what we see when we first activate this pedunculopontine tegmentum, something that selectively activates phasic dopamine burst firing, what we find is it potentiates the hippocampal input without having any impact on the prefrontal input. If we decrease phasic activity, we really don't see much of a change. So this is a system that when it's activated, it potentiates the hippocampal input, but really has no effect if it's deactivated. So the phasic system has to be activated by behaviorally salient stimuli, which is then going to potentiate the hippocampal input. This seems to be mediated by D1 receptors, because we can mimic it by infusing a D1 agonist, and we can prevent it with a D1 antagonist. So the phasic activation of the D1 receptors selectively facilitates hippocampal inputs to the accumbens. Now, what about the tonic system? Well, this we can modulate by manipulating the ventral pallidum. Whenever we inactivate the ventral pallidum, which should increase the number of dopaminergic neurons firing, it doesn't touch the hippocampal input, but it potently downregulates the prefrontal input. Similarly, if we decrease tonic input by activating the ventral pallidum and shutting down dopaminergic neuron firing, now we potentiate the prefrontal cortical input without affecting the hippocampus. So this can work in a bidirectional way. It can increase frontal cortical input or decrease frontal cortical input. And this seems to be mediated by D2 receptors. D2 agonists also inhibit this response. D2 antagonists potentiate the prefrontal cortex. So ventral pallidal inactivation-induced increases in tonic dopamine stimulates D2 receptors and shifts the balance away from cortical inputs. So D1 selectively increases hippocampal drive, D2 selectively attenuates prefrontal drive. So increasing dopamine is going to shift things towards the hippocampus and away from the prefrontal cortex. Now, how does this receptor regulation of limbic and cortical information processing eventually lead to changes in goal-directed behavior? Well, to do this, we do a very simple behavioral task. Unlike Trevor's tasks, we tend to do behavior light. So we do a rather simple task where we have rats that discriminate different types of responses in order to get to a goal-directed behavior. The first is a visual cue task, where we have two arms, one that has a light color, one that has a dark color. And the rats have to learn to always turn towards the lighter colored arm, no matter what direction they're coming from. Then what we do is switch the tasks. So now, instead of paying attention to the color of the arms, they have to make a right turn. So they have to make a turn depending on response direction, ignoring the previous goal of going towards the lighter colored arms. So this allows us to measure how fast they can acquire a task and their behavioral flexibility in changing the task whenever it's no longer appropriate. Now, what we do is a disconnection task. We do this by manipulating the two parts of the hippocampal prefrontal cortical input, but do it in different ways. On one side of the brain, we inactivate the ventral hippocampus. On the other side of the brain, we give a D1 antagonist into the nucleus accumbens. So they're both doing the same thing, but we're still allowing at least one of these pathways to be intact, although now it's modulated in a different way. What we find is that whenever we look at this disconnection, what we find is that the rats tend to have problems both in acquiring the task and they have problems in switching the task whenever we change the salience of the cues. So when the hippocampal inputs are not potentiated by dopamine, there's a disruption in the ability to learn each type of task and furthermore to keep focused on the appropriate response to that task. What about with the prefrontal cortex? Here we do the same thing. We inactivate the prefrontal cortex on one side, and we give a D2 antagonist, or D2 agonist to inhibit the prefrontal accumbens transmission on the other side of the brain. Now what we find is something very different. The rats have no problems acquiring the task, But now whenever the contingencies switch, they can't modify their behaviors. They perseverate. They tend to keep doing what worked before. They're hyper-focused and now can't switch even though it's no longer the appropriate response. So D2 stimulation interferes with prefrontal cortical function. There's a disruption in the ability to switch tasks, and now you get a hyper-focus. So phasic D1-dependent limbic accumbens information processing mediates learning and maintaining a response strategy and goal-directed behavior. Tonic D2-dependent prefrontal accumbens information processing mediates a shifting away from the current response strategy and goal-directed behavior. Under a normal tonic-phasic balance, these systems work together to optimize behavioral strategies. You maintain an effective response strategy by activating and maintaining the hippocampal input, but whenever that's no longer effective, the prefrontal cortex can come in and shift the focus. But if tonic dopamine is disrupted, this would cause a lot more rapid PFC-mediated shifts in strategy even when it's not appropriate. And interestingly, because tonic dopamine works on D2 receptors, if there's decreased baseline D2 receptors, you would expect to have this disruption in D2 modulation. Just like Trevor Robbins has shown that with the ADHD, you tend to get this decreased D2 receptor binding in these limbic areas of the striatum. And interestingly, if you potentiate phasic, for example, by giving cocaine, you should restore the balance. So with the accumbens circuit, the hippocampus keeps the system focused on the task and depends on potentiation by D1 receptors. The prefrontal cortex enables shifting, and this is inhibited by D2 receptors. The normal function depends on an adequate balance of tonic and phasic dopamine. But again, if the PFC is hyperactive or dysregulated, or if there's inadequate tonic down-modulation of prefrontal cortical input because of this decreased D2 stimulation, then there would be an inability to focus on the task, instead being replaced by rapid shifts in contextual attentional focus. In either case, increasing tonic dopamine should restore balance to the system. And this is what we think happens with methylphenidate. If we give methylphenidate, what we're going to get is an increase in this tonic dopamine level, while at the same time down-modulating some of the baseline phasic response. This is going to decrease prefrontal cortical shifting of focus while increasing hippocampal focus on the task. So by preferentially increasing tonic, especially with oral methylphenidate that's going to cause a slow, steady state increase in extracellular dopamine, we should restore this balance in the system. So what we propose is the prefrontal cortex, especially the medial prefrontal cortex, is overly active, either endogenously or by inadequate tonic attenuation of prefrontal cortical inputs, in switching response strategies, preventing contextual focus on the task. By increasing tonic levels, psychostimulants given orally in a continuous maintenance dose should alleviate the abnormal prefrontal switching behavior and allow the hippocampus to maintain focus on the task. An increasing tonic will also decrease random phasic dopamine release, which means stimuli that you should ignore are going to be able to be ignored, whereas the ones that you have to pay attention to are going to be potentiated. And interestingly, the ones that are going to be potentiated most are the reward-related ones, which also seems to facilitate this contextual focus on task. And then just to thank the people who did this work, especially Yukiori Goto, who did the vast majority of work that I talked about, Stan Floresco and Tony West, who did a lot of the work looking at the circuitry of the systems that I talked about, Patricia O'Donnell and Xiaopi On, who did a lot of the work looking at the interactions of these systems. And thank you very much for your attention.
seat and well, thanks to the speakers, you kept tremendously to time. We have good time for discussion. One question, I was really fascinated by your talk. And one question I had is whether it could relate back to some of the things I was talking about, and in particular the sensitivity to salience. So in ADHD you need more things to be more salient to focus. And then the other aspect is, although often people with ADHD have great difficulty focusing and attending, they can flip into a hyper-focused state. And I wondered if your model could possibly explain that. That's one I still find, I see it as a form of dysregulation, that you're one thing or the other, but there's no balance. But I didn't know if your model could explain flipping into hyper-focus.
Actually, I think that from the studies we've done that it does. I mean, I could incorporate some of Trevor's stuff in this because I saw his paper, but I didn't see yours. But what we have is this competition that seems to be occurring between the prefrontal cortex and the hippocampus. And in this case, the prefrontal cortex seems to be winning because of the dopamine dysregulation. But if you drive the hippocampal input enough, it's going to down-regulate the prefrontal cortical input. So ideally, you want the dopamine system to establish the balance. But if you have something that's sufficiently rewarding or sufficiently salient, it can serve to flip the switch itself.
Do you have a comment on that? Or how it might relate to...
I prefer to have some other questions. I think that's an interesting idea.
Yeah. One question I had with your own work, because it's focused largely on impulsivity as a kind of behavioral measure. Do you see a relationship between that and inattention, or do you think it's really the impulsive domain of ADHD that it might tap into more?
Well, I think they're correlated to some degree. So the high impulsive rats are mildly inattentive, but I don't think there's any strong relationship between the two, which is consistent with the fact that probably they're controlled by subtly different networks.
Yep.
So in one case with impulsivity, you're looking at inhibitory response control. In the other case, you're looking at focus of attention. Now, these do come together a bit, notably in the stop signal task, where you've got to attend to stimuli and inhibit at a high level in both cases.
Yep.
But they are dissociable neurally.
Yeah.
And I think they're dissociable probably to some extent by drugs.
Yeah.
So certainly in our animal studies, atomoxetine doesn't do very much for attention. It does a lot for impulsivity, intriguingly. Atomoxetine doesn't do very much for working memory, whereas Ritalin is great for working memory. The problem with Ritalin is that although it may reduce high levels of impulsivity, at the intermediate level, you may actually exacerbate the deficit. So presumably this is why Ritalin is a more successful drug clinically, that it kind of medicates a wide range of deficits. Whereas atomoxetine, has some success, but in our hands seems to be very successful in ameliorating impulsivity in humans as well as animals.
Do you think there's a way forward to predicting who's going to respond better to which drug?
Yeah, well, what I really think would be very useful, other than, for example, measuring dopamine receptor, baseline dopamine receptor changes, will be to focus also on the noradrenaline system. Unfortunately, there aren't many good ligands for measuring basal noradrenaline. I think this is a pity because this has been neglected in human studies to date. I think it does need to be looked at. And I think, Tony's work's brilliant, of course, but we also need to take into account the locus coeruleus and its very important role in modulating cortical function.
So often our concepts follow the technology to a large extent. Edmund, Oh.
No. Yes, it is now. So the default mode network, two aspects I'd like to focus on, both relating to specificity. So of course the default mode network and dysregulation within the networks has been implicated across almost every single psychiatric condition. and beyond. And also the default mode network serves many different functions and has been segregated into functionally different subsystems by Randy Buckner's team, Andrews Hanna and so forth. I wonder if you could talk about what level of specificity in terms of deficits in default network relate to ADHD.
I think in itself, measuring default mode network activity is very non-specific. And probably any, I mean, if you crudely think of things as task-positive networks, where there's an active cortical function, I mean, cortical activity generally relates to downregulation of default mode. As one goes up, the other goes down, and that's largely how the brain works. Although it's interesting that in strategic mind wandering there might be this co-activation, which sort of was an interesting finding that emerged from the mind wandering literature. So in itself, I think it does appear to be a good marker of dysfunction in ADHD, but it doesn't tell you very much about precisely what's causing it. At least that's what I would say in terms of measuring default mode. But it is quite interesting that the literature on mind wandering so strongly implicates a kind of very sort of a default mode which is difficult to regulate as a kind of core problem. But you're quite right, if you read Christoph's paper, and I think I'd find it quite hard to repeat sort of the detailed content of her paper. I probably need to read it a few times more myself. But it rather nicely breaks down the default mode into several functions. And I think they have a general concept, which is that there's a hub, certain hub networks, which underpin internal thought of all sorts. But actually the particular type and nature of internal generated thought is very much down to the different regulatory processes that feed in. And that's where the specificity comes. So I think there's both non-specificity in terms of a generic overactive default mode is linked to internal thought, but actually other hubs and the balance of other systems will give different types of mind wandering. And of course, people like Smallwood and others are starting to say, yeah, actually, you can identify more precisely the different types of mind wandering. So depressive rumination is not the same as the kind of ADHD type of mind wandering. And indeed, most mental illnesses have some form of mind wandering sort of embedded within them. But they do also show this overactive default mode, of course.
Yeah, but I mean, also interesting you mentioned depression. I wonder whether the dysregulation of the default mode in ADHD could actually be a precursor to the development of depressive
Well, I have
problems.
wondered about that. And of course, one of the interesting things about ADHD is you can almost think of it as a very marker of a deficient sort of neural system in terms of the regulation of various processes, but because it's there present from an early age, and then when other factors come along, you have a rather overly sensitive brain that will that can then move on and develop comorbidities like depression. So depression is much more common in ADHD. But in terms of mind wandering specifically, if you have difficulty regulating and controlling your thoughts, if you're subjected to negative life events, you would naturally have a negative thought content. And these are very traditional CBT models, that overexposure to uncontrolled negative thoughts will drive you down in terms of your mood state. So there may be a kind of an interesting mechanism by which unregulated neural networks and unregulated mind wandering can increase your sensitivity and vulnerability to develop depression.
Well, thank you for 3 extraordinarily data-dense presentations. My brain is now full, and I will be driving back to Baltimore as soon as possible. This is a question for Dr. Grace. What I did pick up was interesting in that cocaine increases phasic firing, whereas methylphenidate increases tonic firing. I just wonder if you can expand on that a little bit, because the distinction there seems to be categorical.
So I think a lot has to do with the kinetics of administration. Certainly, if you were to give a bolus high dose of methylphenidate IV, you're not going to get the kind of therapeutic response you're going to get with an oral administration. With the oral administration, what you're going to get is a slow buildup of the tonic dopaminergic concentration that's occurring in the ventral striatum. With cocaine, it tends to be taken more in a rapid bolus. When it's taken in a rapid bolus, it's very effective at preventing the reuptake of dopamine. And because it's going to prevent the reuptake, it's going to strongly potentiate the phasic response. So I think a lot of this has to do with the kinetics of the distribution rather than how the drug is affecting the transporter.
Good, thank you.
Phil, I'm Russ Barkley. This question would be for you, if you don't mind, though if I were permitted a follow-up question, I would have a rather simple one for Dr. Grace. But I found your results intriguing, particularly for those of us who have moved into studying the overlap of sluggish cognitive tempo, or this other attention disorder that is becoming more prominent in the literature, that is characterized by mind-wandering, daydreaming, spaciness, mental confusion, hypoactivity, and a number of other symptoms that are found in the literature to actually form a separate dimension of inattention, that while sharing about 25% of the variance with ADHD inattention, is not ADHD inattention and might more fruitfully be viewed as a comorbidity that can coexist. For instance, our national survey showed that about 50% of people with ADHD have this second attention disorder, and their comorbidity increases their impairment, as you found when you looked at mind wandering overlapping with ADHD. So I'm wondering whether or not your group is looking at now, or maybe plans to look at, and if not, I would encourage them, is the extent to which the mind wandering findings actually pertain to this second attention disorder that, as recent studies have shown, if you don't measure it and don't take it into account, creates some confounding and misinterpretation within the data that you're attributing things to ADHD that really are being attributed to its overlap with this mind-wandering SCT. And I say that because the SCT literature is showing that it has a negative relationship to the hyperactivity, impulsivity, if you co-vary the inattention, but also that it has a much closer affiliation with depression, to some extent anxiety, but more with depressive symptoms and social withdrawal. So I'd love to know if your group is going to explore that, or if you already are. How much is this mind wandering being driven by this other attention disorder, and how much of it is actually inherent to the ADHD that you were describing so nicely?
Well, it's a hard question to answer. When we first started thinking about this, it was based very much on listening to the way patients with ADHD described mind wandering. But you're quite right, there are different types of mind wandering. But it did strike us that there was a very prevalent, very characteristic type, which was really a rather hyperactive mental state, if you like. It was ceaseless. It was a constant flow of thoughts, jumping and flitting and multiple thoughts at the same time, and it kept people awake. And I wondered if that's rather different. It's a question to you, perhaps, that whether that description is rather different from a kind of more dreamy type of mind-wandering linked perhaps to sleepiness or fatigue,
yes,
tiredness.
Right.
The other aspect of ADHD is they tend to have chronic sleep problems. Sleep deprivation is extremely common. So I wonder to what extent sluggish cognitive tempo can be linked to a somewhat different mechanism.
Well, that was explored about a year or so ago by Josh Langberg, because it was an issue that kept coming up as to what extent are hypersomnia or daytime tiredness explaining the SCT symptoms. And he did find that there was somewhat of an elevation in hypersomnia in patients with SCT, but it was not enough to account for the symptoms or the other clinical findings or its correlates. But yes, there was an element of a sleepiness that was part of that, but not serving as a proxy for SCT. So I think SCT remains an important construct. And I agree with you. I think there are qualitative differences in the kind of mind wandering that might be linked to these two, albeit comorbid, conditions. So.
And I think what surprises me about the mind-wandering literature is, although it seems rather subjective, the the evidence points to much stronger relationships to neural activity, if you like, in different mechanisms than you would expect. So it's surprisingly rewarding to think about these things.
Yes, yeah, I would
But
agree.
the answer to your question is probably if we measure SCT in our studies, we can look at
I'd
it in
love to see
covariate
it
and so
if you
on.
would do it. It would certainly contribute to our SCT.
And then you had a question
I have a quick question because several of us are...
You had a question for Tony?
Yeah, for Dr. Grace, just very quickly. Could you explain to us why we're seeing frontal hypofunctionality on fMRI, obviously through BOLD and other measures, and yet you're arguing for almost a hyperactivity of prefrontal systems as possibly explaining some of the SCT, or excuse me, the ADHD symptoms. And so could you help us sort of understand why those two measures aren't necessarily going in the same direction? Is one of them more neurochemical and the other is more bold and functional? Or
Well,
because I'm confused by this.
whenever you look at the functional imaging, there are a number of things you have to take into consideration, and one is which part of the frontal cortex you're talking about. And the part I'm talking about has to do more with the infralimbic, what would be the area 25, as opposed to dorsolateral prefrontal cortex. And I think a lot of the imaging studies have focused more on dorsolateral prefrontal cortex, which has, of course, a much stronger involvement in working memory that tends to be disrupted. And also, whenever you look, you have to consider whether you're looking at baseline or activated states. For example, the schizophrenia literature for the longest time used to talk about hypofrontality because they didn't get an activation of the frontal cortex with certain types of working memory tasks. But once they started looking at baseline, they found that the baseline was actually very high, which is why they didn't get an activation. It was actually hyperfrontality rather than hypofrontality. So I would say that you have to take into consideration which part of the prefrontal cortex you're talking about, and also whether you're talking about an activated state or a baseline state.
Before we come to Tom, I had a question for Trevor, which was one of the things you highlighted, which I greatly appreciated, was the idea that ADHD is the extreme of a quantitative trait. And I mean, as you know, we've done a lot of work on that from family and twin studies, both at the cognitive level as well as the behavioral level. And in a way, there isn't any data that says that isn't the case. I mean, nearly all data points in that direction. But some people then go on and say, and I'm thinking about your work in Cambridge on cognitive enhancement, that Barbara's been involved in thinking about. But some people say methylphenidate has the same effect on everyone. And I've always rather doubted that. And part of the data you presented was saying people, I think you, in your rat experiments, you had rats who were high and low performers, and actually the outcomes of medication effects were very different in the two groups. And I expect you'll see that in ADHD, depending on which arm of the continua you lie on. They're not all moving in the same direction. You'll have different effects at the people at the top end and different effects with people at the bottom end. And I wondered what you thought of that.
Yeah, well, I think in general, we think that Ritalin certainly can improve normal functions, working memory, and attention, and as you saw, stop signal reaction time task, and circular atomoxetine. But there's probably an inverted U-shaped relationship in all of this. And I think that relates to the baseline dependency that I showed you, which kind of captures occasionally this paradoxical effect, which I think is linked to a shift in phasic tonic regulation, whereby the high impulsive animals show a down-regulation of activity and an up-regulation of receptors, and the low impulsive animals show the opposite effect. So, you know, that's intriguing at a subcortical level. So there's probably some baseline dependency effects which relate to this inverted U-shaped function. And similarly, I guess, we know that, for example, if you take individuals who are already performing very well and give them amphetamine or Ritalin, that probably will impair them. And there's data with the polymorphism which shows the same kind of thing. So I think that's what explains it. But basically, I think there's an underlying dimensionality to all of this, which is very important.
I just say maybe...
Because you see, I think the real importance is this, because in terms of causality of the symptoms, you know, I think, for example, changes in cortical grey matter may be really crucial. And what you're really doing is modulating that with these subcortical systems, which are varying in a more dimensional way. So I think that changes the medical model of what one's looking at here. Are you, for example, restoring some lost function, or are you just compensating, improving impaired performance caused by some other damage? And those are the two hypotheses.
And it sounds as if it might be, depending on what you're measuring, some you may get a similar kind of improvement or deficit in the same direction in most people. But for other measures, you'll get more paradoxical effects.
Yeah.
It depends a little bit on your measure.
It depends how well the control subjects are doing, if they're at the top of their game or slightly down.
Right. Thank you. Tom.
These are impressive studies of very complicated processes, but I'd like to ask about two other sources of variation that we see clinically. One is the individual variability of responses, and I think that you'd probably agree that there are differences among rats, and there sure as hell differences among people in terms of the way they respond to stimulant medications. And I'd like to hear a little bit about variability, inter-individual variability, and also, just to compound the question a little bit, in clinical practice we find that there are many people who respond equally well to methylphenidate and to amphetamine, but there are some people who respond preferentially to one or the other of those, a lot better or a lot worse. And I'm curious about how you adapt the information you're providing for us about the details of these processes to the variability factors between individuals, and also how that might change when we're talking amphetamine rather than talking methylphenidate.
Okay, can I answer that? So I think there's 3 or 4 main factors. I did discuss individual variability in my experiments. One obvious individual difference is the baseline difference in behavior. So we had high impulsive rats and low impulsive rats responding in opposite ways to Ritalin. So clearly there's going to be a whole spectrum of altered effects depending on that baseline level, which in turn may be related to basal changes in receptors in the brain, as we also showed to some degree. And of course there are genetic influences which are going to vary across individuals. So you may have, for example, changes in different genes regulating the amines, which are going to influence the response to the drug. COMT, for example, DAT, all of these will modulate precisely what you see, and also the pharmacokinetics and metabolism of these drugs. And on top of that, if you've got individuals who've got varying degrees of grey matter change, for example, for unknown reasons, whether it's genetic or some other environmental etiology, that will also change the individual response depending on the type of task that you're asking them to do. So all of these factors will contribute to the individual variability, and we can but try and capture some of that in these experiments.
Thank you. Anything else from Tony?
Amphetamine and Ritalin is an interesting one.
Okay, please.
There are differences between amphetamine and Ritalin. Amphetamine also affects 5-HT, whereas Ritalin doesn't. And amphetamine has a differential effect on the transporter compared to Ritalin. And Ritalin also releases this recepine-sensitive pool of amine, which amphetamine doesn't. Amphetamine works on the fast turnover pool. So all of these differences, as well as, for example, as Tony said, the route of administration, which orally will make things much slower, whereas IV administration makes things much quicker. And in general, I think amphetamine is much quicker acting, rather like cocaine. So those are the kind of differences that you're going to get between those two drugs.
Yeah, and amphetamine also is a much better releaser than methylphenidate, whereas methylphenidate's a better uptake blocker.
Yeah.
Thank you.
Edmund.
Yeah, so question for Trevor. In our lab, we've been particularly struck recently by the potential role of the amygdala in various delay-related activities. Waiting is quite an aversive experience for people with ADHD. And I was wondering whether you are seeing data implicating the role of the amygdala, in particular weighting impulsivity and temporal discounting.
I don't know whether you've read a paper of ours. The first author is Wynne Stanley. It's published about 12 years ago in Journal of Neuroscience, which showed that in the rats, anyway, that damage to the basolateral portion of the amygdala, which projects directly to the core region of the accumbens, which I did describe, to affect impulsivity, basolateral lesions has exactly the same effect. So I think an amygdala core projection is very importantly involved in that form of weight and impulsivity, whereas actually the premature responding is governed by hippocampal accumbens interaction, probably to the shell region as much as to the core. So it's very interesting, those two forms of impulsivity, although both involving the accumbens, involve subtly different inputs. Orbitofrontal cortex is also important. And by the way, as you saw in one of our early slides, we do see changes in the amygdala structurally in adult ADHD reductions.
Yeah, and that's consistent with what we're finding. Yeah.
Thank
So
you.
I think we'll wrap up in a minute. I was just going to say that's very interesting because the weighting kind of impulsivity you see in adults with ADHD is really characteristic, you know, and if you ask them why can't you wait, it's nearly always because they get irritable. I mean, it's linked to an emotion dysregulation problem, and they feel irritable, and it's such, and even thinking about it is enough to elicit this response. I think that's what you found in your study, wasn't it? Thinking about it altered activity in the amygdala.
Thinking and experience, both thinking and experiencing.
Yeah.
Yeah,
And
it's fascinating that stopping impulsivity and the weighting impulsivity, I think, are governed by different circuits.
Yeah.
And as you showed with Mary Salento in that large study, there's a lack of correlation between those deficits in ADHD. But they're both aspects of impulsivity, which is a multifaceted and probably construct that needs to be dissected further.
I think it was
neurally.
Russell Barkley who's here who termed the phrase emotional impulsiveness,
Absolutely.
which captures it quite nicely.
Beautiful.
Yeah.
So thank you very much. I thought that was a great session and really enjoyed your talks. Yeah.
Learned a tremendous amount from talking. I've got to reintegrate.